Hi everybody, welcome back to another review video. This one is going to be all about transcription. So we're going to go ahead and get started right away, but I do want to remind you that if you are watching this for the first time, maybe you missed school and you didn't see this, then I really want to make sure that you understand how the notes are organized and that your notes end up in the right place. So spend a second and analyze your notes to ensure that you are going to be writing the notes in the appropriate spot. I also want to remind you that it is so important that you listen first and then write. Really digest it, try to put it into your own thoughts, into your own words, and only after you've done that should you write. That's going to really cut down on a lot of your studying time because you've done such a good job learning it the first time. But without further ado, let's go ahead and start talking about transcription. So you're going to see this slide again and again and again because this unit really could be renamed the central dogma. The idea that we move from DNA to RNA to protein. Now, there are some caveats that we are going to see throughout the whole unit, some things that are exceptions, but generally speaking, this is what we see in a cell. We've already talked about DNA replication. That's what this part is showing right here, using DNA to make more DNA. Today, we are going to be talking about transcription, which is making RNA from DNA. Now, you might notice here this star, and that star is indicated over here. That is reverse transcription. And this is possible, but I don't do it. You don't do it. Bacteria don't even do it. The only thing that can really do this that we know of is going to be a virus. So viruses, some viruses, specifically retroviruses, are capable of doing reverse transcription through an enzyme aptly named reverse transcriptase. So if you're asked, what is the major product of transcription? I could totally give that as an easy point on a test. You should tell me RNA. RNA is the product of transcription. Don't get too fancy with it. So that's what we're going to be talking about. How do we make RNA from DNA? Okay, some of the things are the same, and we can use some of our knowledge from DNA replication and apply it to transcription, but there's a lot of things that are different. Luckily for you, when it comes to transcription, there are fewer enzymes that you have to memorize for the AP Bio level. One of the things that is the same is topoisomerase. So topoisomerase is involved in both. It's going to ensure that there's no supercoiling and relieve some of the tension when that DNA unwinds. That's going to happen in both DNA replication and transcription. But there's some different things as well. There's not a true helicase. Now there's helicase activity, of course, because you have to unzip DNA, but it's not a true helicase, or at least not the same helicase as what you see in DNA replication. There's also not a primase. Remember that primase is an RNA polymerase, and we just don't have a DNA polymerase that can start DNA replication. So you have to use primase. You have to use what you've got. So we are already making RNA, so we don't need to have a primer to make more RNA. You don't need a primase. There's also no single-stranded binding proteins that are needed for transcription. So these are just a couple of the surface level things. Let's see more in depth. So in transcription, we really only talk about one major enzyme for AP Bio. Now there are other things involved. There's of course transcription factors. There's other even enzymes involved that we don't really get into detail in AP Bio. But maybe you can guess what this enzyme is going to be because it is making RNA from a DNA template. Well, what made DNA from a DNA template? That was DNA polymerase. So if we're making RNA from a DNA template, we're going to need RNA polymerase. And that's what we're going to talk about. RNA polymerase unzips the DNA at a specific location called a promoter. And most importantly, it adds RNA nucleotides using a DNA template. Now, I want to add a little bit of detail about this first step. So this first step here is a little bit more nuanced than what I told you in class. RNA polymerase in prokaryotes has helicase activity. It is able to unwind the DNA on its own in prokaryotes. In eukaryotes, as far as I know, it actually relies on a transcription factor. A transcription factor is going to work with the RNA polymerase to unzip the DNA. So it's a little bit of nuance. The more important bullet here is the second one. It is going to add RNA nucleotides using a DNA template. And RNA polymerase is just this massive, really cool enzyme that does a really good job. If you want to go a little more detailed, which is definitely beyond the course of this, or beyond the scope of this class, RNA polymerase that we're talking about here is a DNA-dependent RNA polymerase. some viruses have an RNA-dependent RNA polymerase, and that's a pretty fun little fact. Okay, so let's talk a little bit more about this enzyme. RNA polymerase can only synthesize in the 5' to 3' direction. And that means then it's going to sit atop the 3' to 5' strand and synthesize complementarily, right? That anti-parallel structure. So it means that only one side is used at a time. But we have to add a little bit of nuance to this because it's very easy to think, oh, okay, only half of my genome is used, meaning only half of my DNA strand is used, the 3' to 5' strand. That is not true. For any given gene, you will have the template strand and the coding strand. The template strand is the one that RNA polymerase is actually sitting on top of. And so that is going to be the one that is complementary to the final RNA strand, whereas the coding strand is the sequence that is the same as the mRNA, except we replace any T as the U. Now, the coding strand is not used. It is not used in transcription at all. It is just basically for humans to think about and say, okay, if I have the coding strand, then all I need to do is replace the T's with the U, and that is what RNA polymerase would generate using the template strand. Now, the tricky part with this is biologists seemingly haven't come to a consensus on the names of things. And so there's lots of different names that, yes, unfortunately, you are responsible for. So the coding strand can also be referred to as the plus strand, the sense strand, the non-template strand. And then the template strand can also be referred to as the antisense strand, the non-coding strand, and the minus strand. Now, I'm sure depending on what discipline in biology you study, there is more common nomenclature. I've noticed a lot in virology. There is the antisense and sense strand is often used, and the plus and minus strand is often used. You don't typically see coding strand, non-coding strand, and template strand. But if you go into bioinformatics, then I typically hear coding strand and template strand a lot more often. Now, that's just a trend that I've noticed in my master's and my undergrad, but that might not actually hold true. So take that with a little bit of a grain of salt. Okay. Now, I want you to look at this image, maybe even pause and see if you notice anything interesting. I'll give you a second. Okay, so maybe you noticed with gene 1, it is this dark blue strand that is moving in the 3' to 5' direction that is being used as the template strand. But for gene 2, it is the light blue strand, which if we're reading from left to right, that would actually be the 5' to 3' strand. So how is this possible? Well, it turns out RNA polymerase can just flip. It can just turn around. And then it'll synthesize in the direction it wants to synthesize. And so what does that mean? That means that half of my genome is not just wasted. The top strands can be coding for one gene, and the bottom strand can be coding for a different gene. And so it really is gene dependent, and you're going to have to look at the image and really understand which way RNA polymerase is moving based off of the polarity of that strand. So you might be asking, okay, this enzyme seems pretty cool, right? You might be thinking, wow, this is awesome. But how does it know where to start and stop? Well, there is a promoter region. The promoter is a regulatory sequence that is going to be in the DNA strand, and RNA polymerase is scanning the DNA strand until it hits a promoter. This promoter is going to tell RNA polymerase where to bind. So then it's going to bind and it's going to make the RNA, yay, and then it hits a termination site. Same root as terminate, right? So that is going to tell where the RNA polymerase should fall off the strand and end transcription. So those are the two very simple regulatory sequences that tell us where to start and stop transcription. There are other sequences involved that are regulatory and tell a little bit more information. But generally, those are the two most important ones. So let's talk about the promoter a little bit more. So promoters are once again just a region of DNA that have specific sequences. Consensus sequences is what we call them. Consensus meaning that it is in consensus in a variety of organisms. And so, one of the consensus sequences found at the -35 region, more often called -35. So where does this come from? Well, it comes from saying, okay, the transcription start site is here, and 35 nucleotides back is this consensus sequence. And then we also have a -10 region. So that is, starting from the transcription site, back 10 nucleotides, we find this region, which is an AT-rich region. And this varies in prokaryotes and eukaryotes. So don't worry so much about the -35 and the -10. Don't worry about the actual sequences of these consensus sequences. But understand this is what RNA polymerase is looking for. It's looking for these consensus sequences, which we as humans have called the promoter region. So this is the promoter region, just a sequence in the DNA, consensus sequences in the DNA that basically tells RNA polymerase where to bind. This is not the same as the transcription start site. The transcription start site is over here. So the promoter is not the same as the transcription start site. The transcription start site is over here. But the promoter is basically telling RNA polymerase, hey, there's a gene coming up. Get ready to start transcribing. And RNA polymerase says, cool, I will. Okay, so if you're at the end of your notes, I really think it would be wise for you to sort of copy this image down, make some alterations, and make sure that you understand the process of transcription. And so what you should include here is, one, for sure, the very important enzyme that we have talked about today. You should for sure show the DNA strand and the RNA strand that's being created. I would also include the promoter region upstream. And I would show a terminator region downstream, because those are also very important, and it's going to be super important for the next lecture. Okay, so that is everything about transcription. Now, I want you to spend some time and answer these summary questions. So if you can't do this, you probably should rewatch the video and maybe peruse your notes a little bit longer. So I'm going to give you a second to pause, and then I'm going to go over them. Okay, so what is the product of transcription? I sure hope by now you know that it is RNA. You should also know the major enzyme involved in transcription. What is it? RNA polymerase. I'm making RNA, so I need a polymerase to do that. So what is the difference between the coding and the template strands? Well, both of them are the DNA strands, but the coding strand is the one that is not being read by RNA polymerase. The template strand is being read by RNA polymerase, but the resulting RNA strand that is complementary to the template strand is going to be very similar to the coding strand, except it contains uracil instead of thymine. Number 4 here, it says, are all genes encoded on just one of the DNA strands? Certainly not. It depends on each gene, and then RNA polymerase can turn around so that it can still work in the 5' to 3' direction. And then the last one here, what are the 2 regulatory regions that we've talked about so far? Well, one of them is the promoter region. The promoter is going to tell RNA polymerase where to bind. And then the terminator region, which is going to tell RNA polymerase where to fall off. I hope you're doing well in your studying, and I will see you soon. Good luck.